The overall goal of the following experiment is to synthesize an in vivo MRI-detectable apoptosis probe. This is achieved by oxidizing MRI-detectable iron oxide nanoparticles, or SPIO, and conjugating them to annexin protein. Next, apoptosis is induced in adult mouse bone marrow mesenchymal cells that are then incubated with the ANX SPIO probe, which attaches to apoptotic cells. Finally, MRI is used to image the cell culture dishes in order to evaluate the effectiveness of the probe. Results are obtained that show a signal from the targeted attachment of ANX SPIO onto apoptotic cells based on MRI imaging.
The implications of this technique extend toward the therapy and diagnosis of myocardial infarction because early detection will allow for early preventive treatment.
To conjugate Annexin V to SPIO, begin by oxidizing SPIO particles in a solution of 0.15 molar sodium periodate for 1 hour in the dark at 20 degrees Celsius. Incubate the oxidized SPIO with purified ANX protein in 0.15 molar sodium borate for 12 hours at 20 degrees Celsius. To reduce the mixture further, under the fume hood, add 1 milliliter of sodium borohydride, incubate for 1 hour, then quench the solution with 1 milliliter of 0.15 molar tris-HCl. To separate the free ANX from the ANX SPIO conjugates, apply the reaction to a 75 mm centrifugal filter and spin at 14,000 times G